Hi everybody, today I'm going to be going over the delta notch signaling pathway. So just some key components before I get started on the pathway. So we have the ligand and receptor like we've been talking about, an enzyme that's important once it's turned on, and this domain. So the notch receptor is a single pass transmembrane protein. It needs proteolytic processing to function, and how it gets to this proteolytic processing is through the binding to the delta ligand. So the delta ligand will bind to this receptor to initiate proteolytic processing. So that's why it's known as a latent transcription regulator, because it needs to be turned on in order to go into the nucleus to initiate transcription of the gene. And then again, the delta ligand is also a transmembrane signal protein, so it's displayed on the surface of a different cell, so it'll bind to the notch receptor in order to turn it on internally. And then we have an enzyme called gamma secretase, which is intracellular with the cell that has the notch receptor that is turned on. So once it's turned on, it'll cleave the notch tail in order to turn into the NICD, or the notch intracellular domain. Again, so once it's turned on from the ligand, it is an activation gate, so eventually it'll go into the nucleus to initiate the transcription. So why is the notch protein important? It is simple and direct signaling pathway from the cell surface to the nucleus. So once it's turned on by the delta ligand, it'll create the NICD and then go into the nucleus. So it's usually in an inactive state. So once the ligand binds, it becomes active, and it does one proteolytic cleavage naturally. However, once the delta binds, the last two steps of the proteolytic cleavage depend on this ligand binding from the delta, which is important for the signaling process. And another important factor for the notch protein is once it's activated, it's irreversible, so you cannot use it again once you're activated by the delta binding. So how the notch and delta interact with each other, they do what's called lateral inhibition. So it is contact dependent, so they have to interact with each other. So what happens is that the cell in the middle, so the blue one is the neural cell, that develops from an epithelial cell, so it's a precursor cell turning into a neural cell. So you don't want all the cells surrounding to turn into a neural cell. So what happens is that the delta is inhibitory, so it binds to the notch protein on the neighboring cells in order to inhibit it and make sure it doesn't turn into neural, because that is actually lethal for the cell. If all the cells around it become neural, we need those epithelial cells. So again, they bind to each other, and then it makes sure that the neighboring cells do not turn into that neural cell. It's just an epithelial cells. And that's called lateral inhibition. Alright, so this is the pathway that I'm going to go through step by step, especially with the 3 proteolytic cleavage steps. So the first notch protein cleavage, so this just happens naturally, so it's before the ligand binds for the biosynthesis. The notch protein is cleaved in the Golgi to form this heterodimer, and then once it is cleaved, it'll transport to the plasma membrane where it can be turned on once it binds to the ligand. So the second and the third one depend on the binding to the ligand. So this is the extracellular domain, so this is outside of the cytosol. So the signaling cell with the delta will bind to the extracellular notch domain. They have these EGF domains that bind to each other. So the ligand binding actually allows for the conformation change of the notch to allow the cleavage, so it could take this complex away and change the conformation. So it's doing endocytosis. That cell, the signaling cell, is taking up this domain in order to initiate the change in conformation to do that second cleavage. So after the second notch protein cleavage, we have the third, again, depending on the ligand binding. So it's within the transmembrane segment, it'll free the cytoplasmic tail of the activated notch protein. So again, it's going to create that NICD complex, and then the enzyme that does that is the gamma secretase, like I talked about. But once it is cleaved right here, and it makes that domain, it is irreversible. So once it is turned on, the notch tail will actively again go to the nucleus, and it cannot go back into this state. So once you create that NACD from the second and the third cleavage step due to the ligand binding, it'll bind to this RBPSUH protein, and then it will convert. So it was a repressor, it was being repressed, but once it binds, it'll turn into a transcription activator, and then it'll do the transcription of Notch target genes. And the genes are useful. They do cell differentiation, they help with cell fate, it's cell proliferation, so it just depends on whatever the cell needs to do. So since it is irreversible, you can't go back into previous states, there are different ways that the Notch signaling will get turned off. So you can do proteolysis, so you can just completely degrade the NACD domain to make sure it doesn't go into the nucleus to do more transcription. You can degrade the Notch receptor so it doesn't bind to the ligand. Again, turn on the activation gate to make the NACD domain. Or you can have the ubiquitination of the NACD, so, killing the NACD, so again, it can go into the nucleus in order to initiate transcription of the gene. And then here is it more complex. If you look closely, you can actually see the different cleavage steps that I talked about. So the one that happens naturally in the Golgi, and then again, it'll bind to the delta ligand here to do cleavage, and then we have the cleavage to create the NACD domain, and then it'll go into the nucleus and then do all of the transcription for whatever genes that it needs to do the cell differentiation and stuff like that. And yeah, that should be the pathway. Thank you.